Okay, good morning, good afternoon, good evening, depending on where you are in the world. You're joining the study session on transport today. As we wait for everyone to get in, take a look at this first slide here. So while we're waiting to get started, answer these two questions by using the QR code on the left to get the answer form. So the QR code on the left is for the answer form for all of the questions that we're going to be looking at today. And every time you submit a response on the answer form, you have to click submit and then do a new form. But if you could, while we're waiting, answer the question, what country are you in, for number one, and then give a rank from 1 to 10, estimating your knowledge on the topic that we're going to talk about today. So from 1 to 10, 1 being I have not studied it yet, I know nothing about it. 5, somewhere in the middle, I have studied this topic, I don't understand it too much, or between 5 and 10, I only need a refresher and I know this topic well. So I'm going to give everybody a minute to respond to those questions. And I like to see the responses in real time. So please be sure that you press submit, not only now, but as we continue every time you answer a question or give your best educated guess on a question. Go ahead and click on that QR code on the left for the answer form. And I see we have someone from Australia. We have someone from Brazil. I am currently in the Philippines, so we are all over the place. Let's see if we can get a few more responses before we get started. So again, make sure that you click submit every time. Otherwise, I can't see your responses. Okay, and once again, if you're just joining us, we're just going to spend one more minute letting any stragglers in and answering these questions by going on the QR code on the left for the answer form and answering number 1, what country are you in? And number 2, how do you feel about the topic from 1 to 10, 1 being I have no idea and 10 being I only need a refresher. Okay, all right. We're gonna get started to make sure we have enough time to get to everything. So today is the study session on transport. This is the content that everybody has to know, so there's no additional higher level content today. The answer form for all of the questions can be accessed by the QR code on the left here. And the notes that are very much the same to what we're going to be talking about today can be accessed by the QR code on the right. All right, so a few notes about today. So put your content and exam questions in the Q&A section. So if you look, you'll have on the webinar, you'll have a Q&A section, and then you also have the chat. The chat, however, will not be closely monitored. So if you do have any questions, the Q&A is going to be monitored. So please put your questions there. And then once again, when you go onto that answer sheet, it's a Google form to submit the answers to the questions. Please make sure you press submit every time so that I can see your responses in real time so that I know if everyone's with me or not. All right, and these QR codes are going to be on nearly every slide. If I could fit them in there, then I put them on the slide. So don't worry about rushing to get them right up at the start. Okay, so here's a vocabulary list for what we're going to be covering today. I'm not going to read all of these now. This is just for your information. If we have to go back to this and kind of, if we have to revisit this, it's here, or if you would like to revisit this, this is in your notes, but these are all of the words. I should say this is the majority of the words that we're going to be covering today. And then here's a general outline for today. So first, we're going to discuss capillaries and the adaptations that help capillaries perform their functions, arteries and veins, and the same, the adaptations that help capillaries perform their functions. What do these arteries and veins look like in micrographs, and how can you identify them in micrographs? How can you tell the difference between them? How do we measure pulse using the carotid and artery and the radial, and why do we use these? We're going to discuss coronary artery occlusion and coronary heart disease and the incidence data associated with that. And then finally talk about transpirations and the drawings and how you need to annotate those drawings. And within that, I've thrown in some multiple choice questions. At the end, we will have some longer response questions, some extended response questions. All right, so let's start with capillaries here. So capillaries are going to be the smallest vessel that we have carrying blood in the body. So the order of blood travel here, I'm going to use my laser pointer here. So let's say if we have an artery coming from the heart. So I'm looking at this artery in the lower left here. So if we have an artery coming from the heart, arteries are relatively, they have a relatively large diameter. They're big around. So those arteries have to branch into smaller arteries called arterioles. Okay, so in the notes here, we have arteries branch into arterioles. Arterioles are smaller than arteries. And in order to be able to do any meaningful exchange of materials like oxygen, carbon dioxide, nutrient exchange, then those vessels have to get even smaller. So the arterioles are small, but then they branch into even smaller vessels, and those are the capillaries. So in here in the center, this is representing, so we have tissue cells, right? This could be any tissue in the body. This could be muscle tissue. This could be lung tissue. This could be any tissue. So you have a capillary bed that surrounds every single alveoli, right? So this is where the exchange is being done in the capillaries, between the capillaries and the cells of the tissue. This is why any place that you poke yourself or get poked in your body will bleed, because everything is infused with capillaries. Once that exchange, or once the blood flows through those capillaries, the blood's going to be collected into venules, which are like small veins. They are small veins. And those venules are going to be collected into veins, which are larger than venules. So we have capillary bed drains to venules. Venules lead to veins. One of the issues with capillary beds is that the pressure and the velocity are lost in the capillaries. So the veins are going to have adaptations that accommodate that, and we'll get to that in a little bit. And in these capillary beds or capillary networks, the blood, they are so narrow that the blood is forced to travel, red blood cells are forced to travel through single file, right? And that's advantageous. That's a good thing, because then that maximizes the diffusion that can happen between the capillaries and the tissue itself. There, I just have to fix my screen here. Okay. And the other thing that these capillaries provide is a lot of surface area. So everything about these capillaries is going to be encouraging and designed for maximize exchange. All right, so let's talk about some adaptations. We can actually call this capillary adaptations. Okay. You don't have to worry much about the basement membrane here, except that there is one. It's just a membrane around the capillaries. More importantly, though, is you can see in this diagram that the cells that line the capillary, that make up the capillary, are very thin walled. So we have very thin, and they're only one cell, right? So anything that's in this capillary or outside of this capillary that it needs to diffuse into the capillary or out of the capillary only has to diffuse through one cell layer. Now, it's important when you're speaking about this to not say that the capillaries are one cell thick. You want to say that the capillary walls are one cell thick. I'll just kind of squeeze it in there. And it's important to say that the capillary walls are one cell thick because the capillaries themselves aren't one cell thick. Okay, then some types of capillaries that need to have even faster diffusion or maybe need to allow larger molecules through have additional adaptations, and these adaptations are really just larger pores or holes that we call fenestrations. These are just larger holes for materials to move through to allow bigger things through, to allow faster movement. Okay, wait a second. And right, so we have capillaries have very thin walls. They are themselves, the diameter is very thin. And we have, I can add this, and whoops, I'll just squeeze this in here. We have lots of surface area. I know I put that on the slide before, but I'll add it here too because that's very important. Lots of surface area. And now I can't squeeze that in there. Lots of surface. Okay, we'll keep it at that, but it's lots of surface area. Okay, so I like to see if everyone's still with me. So on that survey, on the Google form, the answer form, which is here, if you're feeling good about what we just talked about for capillaries and you're ready for some questions, give me a 1 for thumbs up. If you're feeling so-so, give me a 2 for, thumb kind of to the side. I feel okay. If you're not feeling so good at all about capillaries, give me a thumbs down for a number 3. Or give me a number 3 for a thumbs down. Okay. And I'm just going to wait for a couple more responses. Give you an opportunity to respond. Okay, let's take a look at a multiple choice question. You can attempt to answer this. And again, on the answer form, I'm going to give you 2 minutes or until most responses are back, and then we'll discuss this. So go ahead and read it and attempt, give an answer attempt. All right, I have about half of the responses I want. I'll give you just another 20 seconds here. Okay, good. We're a little bit higher than 50-50 in our response. But so which of the following best describes the effect of a capillary's narrow diameter on the rate of exchange? The correct answer is actually B. So increasing the rate of diffusion by slowing blood flow and increasing contact time. So by slowing the blood and increasing the amount of time for diffusion, we get more diffusion. One way we can see that D is incorrect here. So increase the rate of exchange by increasing blood pressure. Definitely not because of the word increasing. Okay, capillary networks definitely are decreasing the blood flow or the blood pressure. For letter C, no effect on the rate of exchange. The whole system is set up to increase exchange. So that's why we have no effect is incorrect and decreases is incorrect. All right, let's try our next question. I'll give you a little less time to answer this one because there are fewer words, but give this one a try. Okay, good. We have mostly correct responses for this one. So the primary functions of fenestrations in some capillaries is to also B, facilitate rapid filtration of substances. So again, those fenestrations are only present in some specialized capillaries where even a more rapid exchange of materials is needed. So we see these especially in the kidney, where the blood is being filtered and things need to go through rather quickly, and maybe some larger things as well. They do not provide any structural support. I mean, they're in fact larger holes in the capillary. They do not increase the blood flow rate and regulate capillary permeability to gases. No, they're not going to have any effect on permeability to gases. Okay. Question number 3. I'll give you a minute for this one. Okay. This one, mostly correct. Well done. So this one, which adaptation allows for the greatest rate of diffusion of oxygen from the blood to surrounding tissue? The correct answer is C, right? So the short diffusion distance to the thin capillary walls. For letter D, the high concentration of carbon dioxide in the tissues. I like to go from the bottom to the top here when I'm discussing my answers here. So for letter D, the high concentration of carbon dioxide in the tissues, the concentration of carbon dioxide isn't going to have an effect on the rate of the diffusion of oxygen because they're going to be acting independently or diffusing independently. For letter B, a large number of red blood cells passing through the capillary. Yes, there's a large number, but we're talking about a specific like structural adaptation. I know it doesn't say structural adaptation, um, but again, we're talking about like the capillaries and their adaptations. Um, so it's not going to be the large number of red blood cells passing through the capillary, and it's going to be the same. No, it's a different reason for letter A. So the high blood pressure within the capillary, no, the blood pressure is going to be dropping in the capillary. Okay, so we're going to move on to adaptations of arteries and veins now, and then look at some micrographs and how you can easily identify micrographs or microscope pictures of arteries and veins. So first, obviously, this is not, these are not micrographs. This is just a diagram, but for some points on the differences between arteries and veins, I like to start with arteries. When we're talking about arteries and veins, remember arteries, whether they're arteries and veins, has almost nothing to do with whether they're carrying oxygenated or deoxygenated blood. It's the direction of blood flow either away from the heart or back to the heart. So arteries away, right? Those both start with A, arteries away from the heart. It's a good way to remember it. The blood, when it comes from the heart, is under an enormous amount of pressure because it's just been squeezed out of the heart with a great deal of force. So the blood is under high pressure. And so so arteries need some adaptations in order to cope with that amount of high pressure and to also maintain the amount of high pressure because that pressure is necessary. So kind of zoom on this for a moment, right? So arteries, yes, this is a diagram, but all of the features are here. So an artery is going to have a very thick outer wall to help basically keep the artery intact, to be able to counteract that great deal of pressure. There's also called a thick media, but we don't have to call it a media. It's muscle. It's a thick layer of muscle that can regulate that blood pressure and can change the amount of pressure that's put on that blood in the artery. And then the inside of the artery has a very small lumen, which is really just the inside. So the lumen, right? This is just the inside. This is the size of the inside of that artery to maintain that pressure. Okay. Additionally, what's not shown here, there's elastic fibers. So the smooth muscle is shown. The elastic fibers are going to help maintain the structure of the artery under that high pressure. And that's going to be controlled by the autonomic nervous system, right? So we don't have any control over this. Our nervous system is controlling the pressure in those arteries all on its own. Okay, here's a capillary again. We've already discussed that, right? But its adaptations, very small lumen, and the wall is a single cell. Veins, on the other hand, veins have, which isn't shown here, veins have one-way valves to prevent backflow. So once the blood leaves the capillaries, it's going to go into veins, and that pressure is going to be much lower. And what happens as the blood is pushing through, it's kind of pulsing. And between pulses, between forward pushes, because it's under low pressure, it kind of wants to sag back a little bit. It's coming back between between pushes. And so veins have one-way valves that snap shut due to that backflow or the attempted backflow of the blood to prevent the backflow. And that's going to help, that's going to improve the efficiency of the blood moving through. And we have skeletal muscles helps move the blood through the veins, right? So this is one of the reasons if you're on a long flight, you want to get up and move every once in a while because you don't want your blood pooling in your veins because that can lead to clots in some people, right? So looking at a vein, we have a much thinner outer wall because it's not necessary. It's not under a great deal of pressure. So that thick wall is not needed. The large lumen, right? So the inside is bigger as to not like exclude any blood, just to kind of let all of the blood into the vein. And then the lining is media. That lining in that muscle layer is much thinner. And then we also have the valves. So arteries away and veins have valves. And if we're trying to identify these based on micrographs, we have some very clear differences. So let's start with the artery. The artery is labeled there on the left, but the artery has very thick walls. You can see the lumen. It's circular because of the thick walls. It's not compressed easily. Whereas veins are much more variable in the size of the lumen, and you can see easily compressed. So this is the lumen, and you can see it's smushed, right? So easily compressed, but much larger lumen, slightly thinner walls, and you can see this lining. Well, the red doesn't show up very good on the red, but the lining is much thicker in the artery. So that's how you're going to differentiate between arteries and veins in micrographs. Larger lumen, thinner walls, veins are easily compressed. Okay, so same question about arteries and veins and the adaptations of arteries and veins. Give me a 1, 2, 3. How are you feeling about adaptations of arteries and veins? And then we can go on to some questions. Okay, so not too bad. We're about 50-50. Let's try some questions and maybe that could help a little bit. So again, I'll give you a minute and a half maybe to read and respond to this question, and then we can go over it. All right, so it looks like we have mostly correct responses. So the venous return to the heart, so that just means venous blood. Return to the heart is primarily aided by C, skeletal muscle contraction and valves. So elastic recoil for letter D, elastic recoil of the vein walls. There's not much elastic recoil happening in the veins. They don't need it. That's talking about the arteries. The arterial walls uh, can do elastic recoil, the veins not so much. Contraction of the heart's ventricles, again, not so much. I mean, it's going to help a little because there's push behind it, but primarily it's going to be the skeletal muscle contraction and valves. And then high blood pressure, definitely no, because that venous blood is not under high pressure. All right, another pretty short question, so take a minute and give that one a try. Okay, so this one we did pretty well. The elastic tissue and arterial walls primarily functions to, again, C, maintain blood pressure, right? So it can contract and relax depending on the blood pressure that's needed. Reduce friction. We never really said anything about that. That's always ideal in a artery or a vein. You don't want any friction because that can cause problems with tearing. Facilitate gas exchange. No, it really has nothing to do with gas exchange. Prevent backflow of blood. That's really referring to the veins, the valves in veins. So the best answer is going to be to maintain the blood pressure. Okay. One more question here. Give that one a try. Okay, so it looks like almost everyone got this right. So compared to arteries, the lumens of veins is generally A, large and more irregular, which reduces resistance to blood flow. The answer is not D, variably sized depending on the metabolic activity of the surrounding tissue. That really has nothing to do with it. Consistently the same size. They're a bit irregular. Significantly smaller. No, that's the opposite of the truth. So it's larger and more irregular, which reduces resistance to blood flow to make sure that blood is getting back to where it needs to be. All right. Now on to measuring the pulse. Okay, so typically what's done in measuring pulse, we've all probably seen it, maybe have tried it. Sometimes it works, sometimes it feels like it doesn't. But typically we either use the carotid artery in the neck or the radial artery in the wrist using the middle and index fingers, right? Because those are going to be the two most sensitive. The carotid, if you can feel your trachea in front, the trachea has these ridges of cartilage that keep it from being compressed. You can feel your trachea, and then just on the outside of that trachea, if you press two fingers, not too hard, but with a little bit of pressure, you should be able to feel some rhythmic pushback. So that's one way to measure for the pulse. The other is in the wrist. Some people find this one easier, some people find it more difficult. For me personally, I find it more difficult. But feel on the wrist when the palm is facing up. So you take two fingers again and kind of poke around on the sides of the tendons that you can feel there. I can never feel this one, but some people swear by it. So it's kind of measure a preference. What can you do? What do you have practice in doing? I don't have a lot of practice testing for radial pulse, but the reason we use either the carotid artery or the radial artery is because these are arteries that are close to the surface of the skin, so they're easily palpable. We can easily feel them, right? Um, and it'll give like a kind of a quicker feedback than trying to go for something else. Also, they have a bit stronger pulse, so they're not kind of competing with your own pulse that you might be able to feel in your fingertips if you're able to do that. Um, okay, and then I combined this section with coronary artery occlusion because there's just not much to measuring the pulse. Um, so let's discuss coronary artery occlusion. So first of all, when we talk about, let's see, first we'll talk about coronary artery. Okay, so it's tempting to think that the heart muscle itself can just take some of the blood that's flowing through it and use the oxygen and nutrients that's coming from that blood for its own metabolism, for its own um, sustenance, to sustain the heart muscle. However, the muscle, the cardiac muscle, the heart muscle, needs its own supply of blood. So it needs its own capillary network, which means it needs its own designated artery to bring it blood, to bring it oxygen and nutrients. And it's called the coronary artery. So if you've seen a picture of the heart, either real or like a diagram like this, you'll see kind of a lacy structure on the outside. You'll see something else here, right? But this is the coronary artery. This is the artery. These are the arteries that are bringing the heart muscle itself blood. And one of the major causes of heart attack are occlusion to that artery. Okay, so when we get a buildup of plaque, and plaque is made of cholesterol and other things like calcium and different deposits, when we get a plaque buildup inside any artery, but specifically the coronary artery. So here we're showing plaque buildup on the vessel walls. There can be a clot that forms here, kind of caused by the plaque buildup. And that means that the blood cannot flow through here as it should. And we call that occlusion. Occlusion really just means blockage. When that happens, that means that all of the heart muscle that was being supplied oxygen and nutrients through that coronary artery is now starved for oxygen, starved for nutrients, and it's going to die. It's dying, right? And that is what we typically call a heart attack. So I got cut off there a little bit, but heart attack can result when the cardiac muscle lacks oxygen. So that's the heart muscle itself dying because it doesn't have enough oxygen. So there's correlations in this data, right? So it's important to remember correlation is not the same as causation, right? So things that we see in people that experience coronary artery occlusion are increased age, typically is going to happen in more men than women. However, it does definitely happen in women. Genetics, so if this runs in your family or someone's family, they're more likely to have this happen. Diet, some people with diets with high saturated fats or unhealthy diets, there's a correlation between diet and coronary artery occlusion. Diabetes, high blood pressure, high cholesterol, weight, and smoking, right? So these are all correlates. These are all things that we see in people that experience coronary artery occlusion. Okay, I want to talk just a little bit more about this, but it's important to remember that it's difficult for us to determine causation because, first of all, we're humans and we can't ethically experiment on humans and say, okay, all of you people we're going to give you high cholesterol and you people we're not going to give high cholesterol and we're going to see who gets heart disease. We can't do that, right? So we have to look at correlation studies, and many variables are difficult to control for, so that we can't say that one thing causes the other. That's difficult to do in humans. Okay. Okay. So again, 1, 2, 3. How are you feeling about pulse rate and coronary artery occlusion? And are you ready for some questions? Okay. There we go. So let's try some questions here. This one's a bit longer, so I'll wait until I see most of the responses in, so give it a try. Okay, so we did pretty well on this one. The carotid and the radial arteries are frequently chosen for pulse rate measurement primarily because the correct answer is D. They are superficial and easily accessible, allowing for convenient palpation. And palpation just means you can feel it. So anytime a doctor palpates something, they're just feeling it. Okay, so look at letter C. They carry oxygenated blood directly to the brain and extremities, respectively. It's mostly true, but that's not why we use them for pulse rate. They are located close to the heart, reflecting immediate cardiac activity. Again, we don't need immediate cardiac activity when we're looking for a pulse rate. And they're the largest arteries in the body. No, not necessarily, ensuring accurate readings. And even if they are the largest, that doesn't necessarily mean they're the most accurate. Okay. Didn't use the word atherosclerosis, okay, but and give this one a try. Okay, I didn't use the word atherosclerosis, but the primary cause of coronary artery occlusion is the buildup of these fatty plaques, right? So atherosclerosis really refers to a hardening of the arteries, which, if you think about it, that says hardening, sorry about that. If you think about it, these fatty plaques are going to be one cause of hardening of the arteries. Hardening. Okay, because if you have a blockage or the formation of a blockage in an artery, that artery is not going to be as flexible as it was. We want our arteries to be smooth, have a smooth lining, and to be flexible. Because the more flexible they are, the more they can maintain or can respond to and be flexible to changes in blood pressure, especially higher blood pressure. If they are hard, it's going to be more difficult for blood to pass through, and then you could also have breakages, tears of the arteries. Okay, we're not finished yet. However, we're about 3/4 of the way through. We would love some feedback to help us improve the sessions in the future. So over in the lower right-hand corner, there's a feedback form. You can give your feedback. This is a different QR than the other two. Give your feedback. I can give you a few minutes to get that started, and then I'll go on to the next slide. And if you aren't able to finish the feedback form in the couple of minutes that I give you here, you can always finish it either as we're going or when we're done. So I am going to give you about a minute and a half or so right now to fill in that feedback form, and then you will continue on and talk about transport in plants. And then at the end, as I said, we do have some longer response questions. So take a minute and start filling in that feedback form, please. Okay, if you're still working on that feedback form, just give it a pause on that, and you can finish it after we're done here today. But I would like to move on to transport in plants. Okay, so we're going to talk about transpiration and the drawings and the annotations that you're responsible for when it comes to plants here. All right, so first, the first tissue that we have is xylem. Xylem, and here's a micrograph of xylem. We can see that the xylem, I'll just use my laser pointer here because the picture is already red, but the xylem here, we have nice big vessels for the water to move through. So water transport occurs in plants through xylem vessels. The cells that make up xylem vessels are dead and hollow. So when these vessels form, once they're at maturity, the cells that led to the formation of the xylem die. And then what's left over is basically the tubes, the straws of xylem, because all they want to transport, their only function is to transport water. So nothing else is really needed. They're just really a conduit for water to travel through. And you can see that the cell walls here are quite thick compared to the ones around it, and that's because they contain a lot of lignin, which is carbohydrate that's within the cellulose of the cell walls of those cells forming the xylem. So lignin makes it very, well, thick and strong. You can think of it kind of like wood, but not really wood, just very thick and strong. And the xylem is going to appear large. So again, you can just tell from the micrograph, xylem is very large with thick walls on the micrographs. So there's xylem for you. Okay, so how is the water moving up through the xylem? We have the cohesion tension theory, the tension cohesion theory. There's, it's called a couple different names. But to sum it all up, the xylem acts like a straw. And when we think of straws, we think of the great deal of suction that like we can put on a straw to move a beverage up through. And it's very much the same that's happening in xylem vessels in a plant, because we have an upward movement of water. Well, yeah, right. It's moving from roots to shoots. It has to come up from the roots. But there has to be something pulling the water up, right? So when we're sucking a beverage through a straw, we are providing the suction, right? What's providing the suction in the xylem? It's the water lost from the leaves in transpiration. So transpiration is really just the evaporation of water from leaves. And it doesn't seem like it would be enough to pull the water through, but it definitely is. That's the tension part of the cohesion tension model or theory, right? So the tension is being provided from the transpiration, which is sucking the water up from, sucking the water up out of the leaves from the roots. The water tension pulls water up in the xylem, right? And the cohesion part is because for every water molecule, right, these are individual water molecules here, right? They are able to form hydrogen bonds with each other. And if you don't know or remember what a hydrogen bond is, that's just the interaction of the slightly positive to the slightly positive part of one water molecule to the slightly negative part of another molecule. So they're sticking to each other and kind of pulling each other up as they're being sucked up to the leaves. So the cohesion ensures a steady column of water. There can't be any breaks in it. If there's a break in that water column, then that's it. That xylem vessel is dead, basically. But right, the cohesion is the water molecules themselves sticking to each other. Water molecules attracted to each other. And they're also attracted to the walls to each other, right? And that's kind of how they're able to move up. And then the tension or the suction provided by the transpiration, the cohesion tension theory or the cohesion tension model. Okay. Here we have one of the things that you have to be able to do: draw, label, and annotate a transverse section plan of a dicotyledonous stem. Okay. What does that all mean? So let's start with, there we go, the stem, right? So hopefully we know what a stem is. That's the part of the plant that's long, holding everything up. Dicotyledonous, you don't really have to worry about what that means. It's just that the stem is going to look like this. A dicotyledonous plant just means that it's just a way to describe a plant. When the plant comes out of the seed, it's going to have two baby leaves. That's all that means. So di means two, cotyledon, baby leaves. Okay, it's just a way to classify a plant. Transverse section means that if, well, here we go. If I draw a little flower here, you can see my beautiful picture. Wow, okay. A transverse section is going to be cutting it this way, and then you're looking down the stem. So here's a micrograph, and basically what you have to do is look at that micrograph and label it. Sorry, draw your own picture. Doesn't have to be beautiful or perfect, but it has, these things have to be able to, you have to be able to identify these things, label it, and annotate it. Okay, so annotate is the one that students often leave out. You need to label it. So what are these things? And then annotate it. What do these things do? Okay, so what you'll see here is in the stem, you have these bundles. And within each of these bundles, I'll blow one up. I often say that they kind of look like candy corn. If you're not familiar with what a candy corn is, it's like a triangle with softened edges. But in each of these bundles, you have vessels identifiable as xylem, right? So big circles mean xylem. The big circles with the thick walls are xylem. And then over here, you have phloem. The xylem is always going to be more to the inside of the plant. And you can think of it as the plant really wanting to protect its water, but it's just that the xylem is closer to the center of the plant. So this is phloem. And then this is the xylem. Let me back out here. Okay, so the epidermis, that's always going to be on the outside here. Protection and water loss prevention. The cortex, that's all that stuff in the middle that's not xylem and not phloem and not epidermis. Unspecialized, sometimes for storage, xylem for water transport, phloem for carbohydrate transport. And then each one of these, well, now it's getting a little messy, but each one of these bundles is called a vascular bundle, and that's for containing xylem and phloem. So what you would have to do is basically take this drawing and say, okay, I can draw that. And here we go with my drawing skills, right? Okay, not too bad actually. And I try and typically fail to draw the candy corn shape. So my vascular bundles are going to look something like this. And you're going to draw those all around the perimeter, all around the outside. Probably fewer than I'm drawing here because I'm only going to focus in there. Oh no. I'm only going to focus on a few of them, right? So I want to say that this is the epidermis. I'm not going to annotate it now for time purposes, but basically you would write this right underneath your label. So for epidermis, I would write protection and water loss prevention. You're going to have the cortex. And again, the little annotation would be there. For xylem and phloem, so first of all, I would say this whole thing is a vascular bundle. And then I'm going to zoom in on one of these, put big, let's try that again, big circles here, and then I can label that xylem. And again, I would annotate it. What does xylem do? And then little circles over here. And I would label and annotate that as phloem. Okay, so this, my drawing doesn't have as much detail as you would need. You just need to make sure that you include those annotations. Okay. The other thing you need to be able to draw, label, and annotate is a cross section or a transverse section of the same kind of plant, the dicotyledonous root. So here we have a micrograph picture, a micrograph picture of a transverse section, much of the same parts, except obviously the xylem and phloem look different. So I'll zoom in here. Okay, so the xylem, again, easily identifiable, right? You can see these big circles with thick walls, and then you can see little cells over here. That's going to be the phloem. Okay, so and this whole thing here is the vascular bundle. So how are we going to draw this? Okay, so again, I'm going to draw another circle. And okay, this is where it gets interesting. So I'll draw a circle on the inside. That's the vascular bundle. Here's my drawing. Here we go. That's actually good for me. Okay, and so I'll label this as vascular bundle and annotate it, of course. The outside, again, is going to be the epidermis. All of this here is cortex. Okay. And then on the inside, again, xylem is always on the inside. The plant's trying to protect its xylem, so I want big circles. Those aren't as big as I want. Bigger circles. That's good enough. Big circles here, and I'll label that xylem. And then little circles, I'm not going to make all the little circles. Okay, but then you can label that phloem. So there's phloem, and there's xylem. I'm labeling them P and X for time. Now you need to write out the whole thing and make sure you include the annotations. Oh, I forgot the root hairs, right? So also make sure you put some root hairs out here. Again, this is my art. Hopefully yours is a little better, right? Epidermis with root hairs for increased surface area. Okay, so are you happy with plant transport structures? Are you ready for some questions? Give me a response, and then we'll try some questions. All right, great. Almost everybody's still with us, so take a moment here and give this question a try. Okay, good. It looks like almost everyone has that one. So the tension cohesion or cohesion tension theory explains, and yes, the movement of water in xylem. This A is just wrong. It's not talking about sugars. The process of root pressure. I don't know what root pressure is. Opening and closing of stomata. Again, just wrong. Okay, so it's the movement of water in xylem. Okay, another one. Give this one a try. All right, so this one is kind of inadvertently tricky. Like I didn't mean it for this to be tricky, but now that I'm reading it, yeah, it is quite tricky. So the epidermis of a dicotyledonous root is responsible for, and I can see why a lot of you would pick providing structural supports. However, the root itself, the epidermis, it doesn't need a lot of structural support. Like, yes, it does need to keep things inside, and the root is going to be pushing through the soil. More importantly, however, the epidermis here, right? The epidermis grows root hairs. And I mistakenly made that kind of an afterthought in the diagram. And those root hairs are going to be responsible for absorption of water and minerals. Um, this is a question that I wrote. I think anything on the IB test would be a little less ambiguous. But it's a good question to highlight that the epidermis is going to be growing root hairs for increased surface area for water and mineral absorption. Okay, so keep that one in mind. Okay. And question about lignin, give this one a try. Okay, this is another inadvertently tricky one because we're kind of split into thirds here. And maybe it's because I don't recall if I actually said this, but the lignin in xylem cell walls contributes too. So if you're looking at a xylem vessel, and I'll just draw it for now, right? You're looking at a big vessel, but it also has really, let's see if I can do this, thick walls, right? And it has to have those, no, let's just leave it at that. It has to have those thick walls because of the tension that is the tension force that's being put on the inside. So if you've ever had like a really thick milkshake and you're trying to, suck the milkshake through the straw and then the straw just collapses on itself. If the walls of your straw were thicker, you wouldn't have that problem, right? So the lignins in the xylem walls make it thicker to be able to resist the tension caused by the water being pulled through. Okay. So flexibility, no, we're kind of looking at to the opposite of that. Water storage, no, the cell's dead, is not storing anything, and there's no place to store water in the lignin itself. Active transport of minerals, again, this stuff is dead. Xylem is dead, so it's not doing any active transport. Okay. So again, thank you for coming. I'm going to give you another minute to fill in this feedback form. Actually, I'm going to erase my little arrow there and I'll put a, no, what was that? Okay. Um, I'll give you another minute to fill out this feedback form to tell us how we did. And then I have a couple of extended response questions after this slide. So please take a few moments, fill out the feedback form, and then we can go on to the extended response questions. Okay, thank you very much. Again, if you haven't had a chance to finish the feedback form, please do so when we're done. Okay, so this is how I would like to do this one. So the question, explain the structural adaptations of capillaries that optimize the exchange of materials between blood and surrounding tissues. It's fine if you don't have a complete answer, but for the form, for the Google form, please just put down whatever you have. Even if you want to submit one point at a time, you think of something, type, submit, that's fine. But I want to give everybody a couple of minutes to um, share what they come up with for points, for marks in the response to this question. I'll share some of them and then I'll give you the mark scheme. So please take a few minutes and write down and type in to the survey what you have. I'm just writing them in as they come in so you can see my Terrible handwriting there, but if you keep entering in your responses, I can write them up here. Okay, I've summarized what you've all put in here. So let's see, we have 5 here. Let's see what we have in the mark scheme here. Okay, so we have 1 cell thick, and then to allow shorter diffusion distance. Ugh. Fenestrations to allow for faster filtration. So that's 4 marks already because we have the structural adaptation and then explaining how that structural adaptation supports the function. Okay, we have small lumen, more contact time, right? Dense network to make increased surface area. So 1, 2, 3, 4. 1, 2, 3, 4, 5, 6, 7, 8. I'd give that 8 marks. Let's see what we have. Okay, so for large surface area, right, 2 marks. Extensive branching, increases surface area, maximizes contact. Narrow diameter, reduces diffusion distance, slows the blood flow, increasing the time for exchange. Thin walls, composed of a single, thin walls of a single layer of cells. You don't have to put in endothelial cells. Minimize diffusion distance, and then fenestrations, allows for rapid filtration. Okay, so as a group, I think we would have gotten full marks for that one. Let me clear. All right. Next one, again, same kind of format here. So explain how the structural differences between arteries and veins relate to their respective functions in blood transport. Okay, good. Um, I've written down some of what you responded with, right? So arteries have elastic walls to contain blood pressure without breaking. I just added the word flexible. And then I've also added in red, veins do not. So when it says the structural differences, it's kind of like they're asking for the differences. They're kind of asking for a contrast as well. So they want you to to show them that you know that arteries have this, but veins do not. So if you have the time, just write it out, spell it out for them. Arteries have this, veins do not. Veins have this, arteries do not. And that helps communicate that to them, to the examiners, that you know the differences and you're not just kind of um, regurgitating something that you read in the book or a book. Arteries have thick, smooth muscle to control blood pressure. Veins do not. Veins have a larger lumen to allow all the blood in. I wasn't sure how to rephrase that. So for blood collection, so the large lumen allows all the blood in without excluding any because of the large, because of the low pressure. Have a larger lumen. So that kind of implies that you're saying the veins have a larger lumen over arteries. So I didn't add arteries do not in that one. And then veins have valves to prevent backflow. Arteries do not. Okay, and then what does the actual mark scheme say? So thick walls with, da, 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 da. Okay, so I just kind of put the function for all of these things for arteries. I like how we wrote it better than how I have it typed here, actually. Um, it's not wrong, but I still like how we did it over here. Okay. Do low pressure, prevent backflow. All right, let's just go on to the last one. Okay. Discuss the challenges in establishing a causal link between lifestyle factors and coronary heart disease, which is a coronary artery occlusion, using epidemiological data. Using epidemiological data just means you're looking at the numbers of people who have this or who have experienced this and what their lifestyle is like. So what's their age, their sex, their diet, do they smoke, all of this. And the other thing I want to say before you give this one a try, the command term discuss, um, ah, offer a considered and balanced review. So balanced. So you want to make sure you're looking at not necessarily arguing both sides, but saying, without giving away the answer, yes, these two things go together because we know XYZ, but do they really go together because XYZ? Okay, so just take a few minutes and we'll finish this one up and then I'll show you the mark scheme. Okay, I think we've kind of trickled off on our participation here. So I'm going to go ahead and put up the mark scheme that I made for this one. Okay, so supporting the causal link, we have the link is statistically powerful, like we see people. Good, yeah, we can experiment on humans. It can be influenced by many things, both good, right? So for supporting it, we have the link is statistically powerful. We definitely see these correlations. We see the same relationship found all over the world, different places, different studies. It makes biological sense based on what we know so far. And it's not contradictory information, right? Which is kind of the same as makes biological sense, but we don't get a lot of contradictory information. At least we don't have that much in the literature that's well known. And why such a challenge to establish this causal link, right? Correlation does not equal causation. There's a relationship, but not a causal relationship that we know of so far. There's lots of other confounding variables that may influence coronary heart disease. ethical limitations, right? Can't experiment on people, and the reliance on observational studies, which contain lots of things that we cannot control. Okay, so once again, thank you for your participation. And also, once again, if you have not had a chance to fill out the feedback form, please do so. Thank you very much for coming, and I hope you get a chance to attend um, any other um, sessions. Right, thank you very much, and I'll leave this up for a minute before I close the webinar. All right, I'm going to be closing the webinar now. Thank you very much for coming, and enjoy the rest of your day.